Chapter 15, Section 3 is created by Kassidy or Julia Willason, Claire Wess, and Kristina Gallucci.
Linked genes tend to be inherited together because they are located near each other on the same chromosome. Each chromosome has hundreds and thousands of genes. Linked genes are located near each other on the chromosome, and they tend to be inherited together. Geneticists followed linked genes in breeding experiments, and the results deviated from those expected from Mendel's law of independent assortment. Morgan's Drosophila experiment shows linkage between genes and how it affects the inheritance of the two different characters. In this example, it shows a cross between two flies to show their color and wing size. There are two phenotypes to each type of fly. There is a wild type fly with a gray body and normal wings that looks like this. And then there is a double mutant, black-bodied, vestigial winged fly that looks like this. The mutant fly has recessive genes to the wild type, and it is an autosomal cross. Morgan carried out this cross shown, and the P generation to the F1 generation produced a heterozygous, gray-bodied, normal-winged fly that looked like this. He then went to test cross that heterozygous fly with a double mutant fly. The results would show as this. The female heterozygous gray-bodied normal-winged fly and the mutant fly that was the male would produce these four possibilities for offspring. The results showed with 965 of the flies being gray-bodied normal flies, 944 being black-bodied vestigial wings flied, and 206 being black-bodied normal wings, and 185 being gray-bodied vestigial wings. Now, if the genes were located on different chromosomes, the ratio for the predicted cross would be a 1-to-1 to 1-to-1 ratio. But if the genes were located on the same chromosome and the parental alleles were inherited together, it would be a 1-to-1 to 0-0 cross. And that is almost what had happened from the results. Morgan had then concluded that the body color and wing size are usually inherited together and are genetically linked and on the same chromosome. However, since non-parental phenotypes were produced, it indicates that some mechanisms occasionally break the linkage between the specific alleles of genes on the same chromosome. Genetic recombination helps explain the production of offspring with traits that differ from the parent.
Meiosis and random fertilization both generate genetic variation among offspring. During Mendel's studies, he noticed this genetic variation by seeing that some offspring do not have the same combinations of traits as their parents. For example, if a pea plant with yellow and round seeds that is heterozygous for both seed color and seed shape was crossed with a pea plant with green and wrinkled seeds that is homozygous for both recessive alleles, their offspring could have yellow and round seeds, yellow and wrinkled seeds, green and round seeds, or green and wrinkled seeds. Half of the offspring are expected to inherit a phenotype that matches either of their parental phenotype. These offspring are the parental types. However, there are also two phenotypes that don't match the parental phenotypes, and these are the yellow, wrinkled, and green round seeds. These are the recombinant types, or recombinants for short. When 50% of all offspring are recombinants, geneticists say that there is a 50% frequency of recombination. A 50% frequency of recombination is observed for any two genes that are located at different chromosomes and thus cannot be linked. In this example, those two genes are the genes that code for the color and the seed type. The basis of recombination between unlinked genes is the random orientation of homologous chromosomes at metaphase 1, which leads to independent assortment of the two unlinked genes, which is shown by the four possible combinations in this example.
Now let's return to Morgan's fly room to see how we can explain the results of the fly test cross illustrated in figure 15.9. Recall that most of the offspring from the test cross for body color and wing size had parental phenotypes: a gray body with normal wings, or a black body with vestigial wings. That suggested that the two genes were on the same chromosome. Since the occurrence of parental types with a frequency greater than 50% indicates that the genes are linked, about 17% of the offspring, however, were recombinants: a gray body with vestigial wings, or a black body with normal wings. Faced with these results, Morgan proposed that some process must occasionally break the physical connection between specific alleles of genes on the same chromosomes. Subsequent experiments demonstrate that this process, now called crossing over, accounts for the recombination of linked genes. In crossing over, which occurs while replicated homologous chromosomes are paired during prophase of meiosis I, a set of proteins orchestrates an exchange of corresponding segments of one maternal and one paternal chromatid. In effect, end portions of two non-sister chromatids trade places each time a crossover occurs, so at the end of meiosis, you would have recombinant chromosomes. Figure 15.10 shows how crossing over in dihybrid female fly resulted in recombinant eggs and ultimately recombinant offspring in Morgan's test cross. Most of the eggs had a chromosome with either the B+ and VG+, or BVG, parental genotype for body color and wing size, but some eggs had recombinant chromosomes, either B+ VG or BVG+. Fertilization of these various classes of eggs by homozygous recessive sperm, the BVG, produced an offspring population in which 17% exhibited a non-parental recombinant phenotype reflecting combinations of alleles not seen before in either P generation parent: the gray body vestigial wings or black body with normal wings.
Genetic variation for natural selection occurs when new combinations of alleles are created. The behavior of chromosomes during meiosis allows for genetic variation in the offspring. Maternal and paternal homologs are randomly recombined when crossing over occurs in prophase I, and when the homologous pairs line up independently of other homologous pairs, allowing independent assortment to occur during metaphase I. Also, Mendel's experiments demonstrated that another factor of genetic variation is caused by the behavior of genes. All these ideas show us that the recombinant chromosomes come from the new combinations that are created during crossing over and independent assortment during meiosis. Random fertilization, which is any sperm cell that is combined with any egg cell, will further increase the possible allele combinations in the offspring. All this variation provides the foundation for natural selection. If the organism has traits that are better suited for the environment, the organism can be expected to thrive in the environment and later produce offspring, ensuring the continuation of their better suited genetic complements. These alleles will continue to be shuffled into different variations each generation. On the other hand, if the genes are not suited for the environment, the organism is expected to die, thus no offspring can be produced. Finally, the interrelationship between the genotype and the environment determine the genetic combinations that will continue through future generations in the offspring.